Yes, this is a bit of an update just on the rust situation. So pulled together some information from Will Cuddy, etc., out of the Rust Laboratory, University of Sydney, based at Cobbitty there. So just quickly, it's a bit of a background last year. There wasn't a huge issue with rust last year, like there was certainly a bit of it around. Fungicide with the in-crop weed spray has been pretty common, you know, with that mid-tilling up to growth stage 30 stage, so that's keeping a lid on it. That tends to be matched to varieties, but you know, even it's going in with Gregory at this stage. Certainly stripe and leaf rust were evident in unsprayed trials. We monitored 2 unsprayed MVT trials, one at Spring Ridge, one at North Star. North Star didn't develop rust, but Spring Ridge developed quite high levels. And we had another one at Tamworth. Now this was all natural inoculum. So the point I'm trying to make there is rust hasn't gone away. If it's not controlled, the spores aren't the limiting factor. Okay, in 2014, I guess most of the inquiries around stripe rust in Spitfire was probably 90% of what come in. We knew that Crusade and others were going to get it. That certainly dropped off. What we've shown, or what Cobbitty's shown, sorry, is what went on with Spitfire. It was actually off types in the Spitfire. So I did 3 separate pathogenicity tests through the glasshouse in Cobbitty. There is no new pathotype that's taken out the major gene, which is YR29. So all stripe rust genes are called YR for yellow rust. There is no pathotype taking out YR29. So it's actually off types in the Spitfire, and we saw that as individual plants with a lot more stripe rust on them, and then ones around them with a lot less or none. So that's indication of a purity issue. So what it was replaced with really is issues around stripe rust in Suntop. Did anyone have issues with stripe rust in Suntop up this way? It's more back around that Narromine, and certainly as we headed south. And there was one crop on the plains quite late with Lancer got a bit of stripe rust in it. Okay, and that's been the main issue in 2014. I'll talk about that. The thing to do too, so the original stripe rust that come in from Western Australia in 2002, Western Australia got to us in 2003, was the WA pathotype. It's mutated to take out the YR17 gene. That happened in 2006. And then around 2011, we got a combined virulence for the 2 genes, YR17, 27. Okay, this is now established across the region, this pathotype. Okay, YR17 is still the dominant pathotype which comes in off surveys, but this is now established right up into Queensland. The main implication there, anyone growing Livingston, it relies on these 2 genes. You need to manage it as if that pathotype's there. So up this stage, we knew that the pathotype was only down south. It takes a while to spread up north, and we're adjusting our advice each year. Now you consider this established, and you need to manage your Livingston. And there's 2 new leaf rust pathotypes, one that was around last year in Naparoo but showed up again in Naparoo, plus there's a new one out of South Australia I'll make you aware of. That's not to get alarmed and start spraying everything for leaf rust. We just need to be aware that that's developed in South Australia and has the potential over the next couple of years to move up to us in the north. But that should take a couple of years is what usually happens. This is a picture of mace, unsprayed in our trial at Tamworth. This is all stripe rust, taking out the whole leaf. Mace is very susceptible to stripe rust. Mace makes up 60% of the Western Australian crop. 70? What is it? 70%. Sorry, I'll adjust my talk. I'll get it right the second time. Okay, 70. You're with AGT then, are you? No? No. They usually gloat about it. Okay, so let's say it's between 60 and 70% of the crop in Western Australia. So the thing with that is that original pathotype that come into Western Australia hasn't actually mutated in Western Australia. It's actually died out. They can't find anymore. So it has got resistance to the original WA, but this is what would happen if that YR17 pathotype got into Western Australia. This is how susceptible mace is. It's probably about 40% of the crop in South Australia. They have to spray it to manage it, so it's a very susceptible variety. Just pointing out that, you know, it's certainly around. I rated that trial on a 1 to 9 scale, so 1 is where we can't find any pustules, 9 is where, you know, it's getting up there towards the mace, the whole thing's pustule. You can see your range of varieties here. The point I wanted to make here, so this is around the Suntop. So this is the NVT Suntop source. Very limited number of pustules. So it only, you know, averaged a score of nothing or just a few limited ones. This Suntop 1 and Suntop 2, that was a Suntop seed source from Wellington, 2014, where he developed alarming levels, or you know, what he considered alarming levels in the crop. So we actually tested that seed source and put it in this trial. You can see in our trial, we couldn't get any rust to develop on it at all. Okay, and I'll go into why we think that is. And we know this was the YR17 27 pathotype. We have it confirmed through Cobbitty, but Wagon here's a giveaway. Okay, it's susceptible to the YR17 pathotype, but it gets taken out by the 27. So once you lose that, that's what wagon looks like. Not that we grow that. Okay, stripe rust in Suntop, 2014. As I said, it was around in limited situations in 2013 as well. Let's go back a bit of history. So Suntop's actually been tested in NVT stripe rust trials. So these are dedicated, inoculated trials run under high misting to generate the disease. So it's been in 62 trials since 2010 to 2014. This is across the country. In 62 of those trials, it's tested moderate resistant or better. Okay, only once at Canowra 2013 did it actually rate us an MS. Okay, so it's performed pretty well. So it doesn't look like it's a misclassification. There were limited reports Wellington 2013 and certainly several in 2014. As a general, it was the higher rainfall zones, and it was also where they were trying to correct this perception around reduced protein achievement in Suntop. So it's a high yielding variety, but you get that protein dilution because of that high yield. So they pumped it up with N to try and correct for the protein. So the high rainfall irrigated, and generally they were under high nitrogen status is where it tend to happen. Okay, I had a theory running in season that might have been a pathotype issue, so that YR17, 27 pathotype spreading up north. Suntop relies on YR17 and 27, they're the major genes, so those are the ones that stop infection totally. And then it's got an adult plant resistance gene, YR31. So adult plant resistance only kicks in as the plant ages, and it kills off the cells around the pustules and makes that infection non-viable. Okay, so I had a theory, well, this is the YR17, 27 spreading into the north, therefore Suntop's only reliant on YR31, that's why we're seeing a bit more in Suntop. 18 samples of Suntop were sent into Cobbitty from last year. Only 2 of them come out of the north. If you consider Narromine the north, there was nothing further up. And this is the breakdown of the pathotypes. 10 of them are actually the YR17 pathotype. So Suntop should have been protected by 27 and 31. Only one of them, which is one of the ones from Narromine, was actually this one which took out 2 genes. So then it would have been reduced to only YR31. And 7 of them are yet to be identified. They're being purified enough. So my theory is not supported very well. We know it's not related to seed purity. So the stuff from Wellington 2013 we sent off through grain growers. They've got a molecular test. They run 20 markers across it. Very detailed, very accurate way of telling. We know that the stuff from Suntop didn't have impurities. It was 99.9%, which is the most they'll give you, Suntop, and it was definitely Suntop. We tested the Narromine stuff as well. It's pure Suntop. And it didn't really match up with a seed purity issue in the paddock. It tended to be bigger patches, so hot spots, where every plant was getting some level of rust on there. So what's the wash up? Basically Suntop, Spitfire, they largely rely on adult plant resistance genes. So as we get these pathotypes come in, taking out some of their major genes, they're reduced to their APR genes. Okay, Gregory's relying on an APR gene. YR7E is its main gene. We go back, I think it was about 2011, we saw a lot of rust early in Gregory, and there was a furor through the industry about Gregory. The thing with adult plant resistance genes are they're not expressed to seedlings. So they kick in as the leaf ages. So you can get infections there early. And these guys are going to be the same. Okay, and you're going to see a lot more of this. Breeding companies, as a general rule, are moving away from incorporating major genes, YR17, 27, things like that. They break down too quick. Okay, since the original WA pathotype coming in 2003 across to us, there's been 6 mutations. Not all of them been significant, but 3 of them have. We've had the Jackie, the YR17, the YR17 plus 27. And actually there's another one that combined with Jackie, there's 4 that are important. So they're not as durable. So APR genes are called slow rusting genes. They actually just stop the build-up. The plant itself then kills off the infection. So they're not going to be overcome as easily by mutations. The other thing is we know high nitrogen delays the expression of some of these APR genes. Delays maturity a bit, creates a more humid environment as well, more favourable for rust, but actually delays expression of some of these adult plant resistant genes. They're linked to the nitrogen status in the leaf. Certainly cooler wet conditions will favour rust and also reduces APR expression. So there's still that vega around them. So what are we going to do for 2015? So certainly Suntop Spitfire, I think it was adopted with Spitfire 2014. Most people put one spray on their Spitfire. But certainly if you're chasing high protein or got high N status coming out of a good favourine crop, I would be managing rust early in these varieties. Okay, so you can look at it up front or you can look at putting it in with that early herbicide spray. Might be a bit different depending on your summer survival out your way, Andrew. Certainly then monitor and control from growth stage 30 to 39 onwards. Okay, so just react to what you see. So it's great you ring us up and say there's something funny going on my Suntop, but we then need to look at it and actually say manage that and we'll figure out what's going on later. Don't just sit there and watch the rust. Okay, that's stripe rust. Now we're switching, okay, we're going into leaf rust. Okay, it's still a rust but a different species. So this is the pathotype that was around last year and the year before. That's the fancy number. It just combines the genes. All the genes are called LR genes in leaf rust, stands for leaf rust. This is a mutation which combined resistance for 3 genes. First detected Naparoo, Inverell, Warialda, 2013. It's present in Naparoo everywhere last year where it wasn't sprayed. Okay, so that was around Corindi, out to Purley-Wall, around that area where they got the dual purpose. So this is a dual purpose variety grown and grows. Okay, so certainly that's been issued. So anyone growing Naparoo? Anyone got clients growing Naparoo? Probably not, up this way. The key message there is if you've got clients with Naparoo and they didn't know they had to manage it for leaf rust, then they obviously don't have a registered seed source. Because AGT put a thing out, mail out to every registered Naparoo grower, alerting them of this problem, so they should have been aware of it. So hopefully that's not new to anyone. Okay, this is the one that come in. Oh, sorry, the point I should have made out, this is a mutation. So this one arose from mutation of established leaf rust pathotype. The one I'm going to talk about from South Australia is a bit different. It's actually an exotic introduction. So it hasn't mutated, it's come in from outside. They haven't figured out which country it's come from yet, but they're doing the molecular work on that. Okay, anyone into pub trivia? It's the 12th documented exotic incursion of a rust, so that's across the stem, leaf, stripe, in cereals since 1922. The 12th since 1922. That'll be a question for sure tonight. I'll make sure it is okay, and then you know who's listening. Okay, here's the fancy number, combining virulence to a number of genes. First detected in South Australia on revenue. August 2014. By October, it's actually widely distributed through South Australia. They're finding this at low levels where they were looking. Didn't get into Victoria and hasn't been recorded up into New South Wales as yet. Okay, don't get alarmed. These are the varieties from the testing. So there's 2 real good tries that Grant Holloway had at Horsham, where he happened to get hold of this pathotype out of South Australia and did inoculate in Victoria. So it was in Victoria, I lied, but it was inoculated by Grant. It was deliberately a late nursery, so it didn't threaten industry. God, I shouldn't have said that. Anyway, this is what the rough initial ratings, okay? So these are preliminary ratings. There's limited data, but these are the ones that didn't look so flash. Okay, Impala, expected to be an S to VS. It was already S anyway to the pathotypes that are here. And you can see some going down this way. So Suntop, certainly not dropping a lot, but to keep an eye on. Beaufort, we shouldn't be growing anyway because of stem rust. You can see some of these varieties for us. Spitfire is already MS, so it's probably not going to change. You know, it may go down a bit, but yeah, just to keep an eye on. So it's not to get alarmed at the moment. They're provisional only. These were disease nurseries, so they were inoculated and certainly irrigated with risers in the crop, created the conditions, so it's the ideal conditions. And the point I want to stress here is we have had existing leaf rust pathotypes in the north for ages. And we've got varieties there that, you know, Impala is susceptible to leaf rust. Spitfire's been out there and it's been MS the whole time. So how many people have been spraying for leaf rust in Spitfire in the last couple of years? Okay, so just remember that and keep that in perspective. We certainly don't want the suckers if that happens. It will probably take a couple of years for it to spread. So if you go back to the stripe rust, the YS17 pathotype first detected down south 2006. It was 2010 until we saw it up in Queensland. And we saw Crusader drop over, which was quite susceptible to that pathotype. So it should take a while for it to come up to us, but I think we need to start monitoring and that list of varieties that were there before. The other thing is it can get confused. So we had a crop of EGA wedgetail on the Liverpool Plains last year. We expect, you know, sample come in. We thought, oh, it'll be stripe rust. It always gets stripe rust. End up actually being leaf rust. So stripe rust, you always see these characteristic stripes. They tend to be yellow in colour, whereas leaf rust is more scattered. The other thing, I don't know how it comes out, you actually get sort of a green halo around the leaf rust pustules, and you'll see that before the pustules actually come out. So if you look in the lower canopy, you'll see these little clear light green circles. That's the leaf rust before it actually comes out. None of the others you can visibly see anything till the pustules come out. So certainly just to keep an eye on that, and if you have any concerns, then ring us up, and that's the acknowledgement.